Hello, welcome everyone. So this is Bio Seminar Series. The speaker for today is Dr. Upinder Singh Bhalla. Let me give a brief introduction to the speaker today, and then the speaker will deliver the talk. Dr. Upinder Singh Bhalla is a computational neuroscientist, academic, and a professor at National Center for Biological Sciences of the Tata Institute of Fundamental Research. He is known for his pioneering studies on neuronal and synaptic signaling in memory and olfactory coding using computational and experimental methods. He is an elected fellow of the Indian Academy of Sciences and the Indian National Science Academy. He is a recipient of Shanti Swarup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards, bestowed by the Council of Scientific and Industrial Research in 2007 for his contributions to biological sciences. In addition to that, the Infosys Science Foundation awarded him the Infosys Prize 2017 in Life Sciences for his pioneering contributions to understanding of the brain's computational machinery. Dr. Bhalla, I kindly request you to deliver the talk.
Yeah, thank you very much, Shyam, for inviting me, and thank you to everybody who is on this call for coming to hear me speak. And I enjoyed a little tour through the Krea website. It looks like a very interesting and exciting place to be, and I can certainly say that the kinds of breadth one gets through a good liberal education is something which is of incredible value. You find out in years to come what it's like. So it's a great pleasure to speak to all of you. So what I will try to do now is to share my screen, and should all go well, I can begin my talk. So can you see my screen?
We can see your screen, yeah.
Okay, great. So yeah, so welcome everybody, and I will talk about memories and butterflies of the soul. Just a couple of words, as Shyam has so kindly told you, I work here at the National Center for Biological Sciences, and it's a pretty little campus. And if you should happen to drop by, it's a really nice, it's a very intense place where we do lots of exciting stuff, and I will tell you a little bit about that. So where did the butterflies of the soul come from? It came from one of the founders of neuroscience, Santiago Ramón y Cajal, who was actually quite a character, quite an author in his own way, in addition to being an artist, both in terms of his science and in terms of the illustrations that he did by himself of his science. And what he looked at, as you probably all know, is he was the one who really convinced people that neurons were individual cells, and he drew and examined and studied a huge amount in all sorts of animals. And here are just some sample cells that he drew. And he was so deeply into it, he wrote about these. He said, these are the mysterious butterflies of the soul, the beating of whose wings may someday, who knows, clarify the secret of mental life. So that's very poetic. And as you can see, the cells themselves are really a work of art, both in terms of how he drew them and in terms of how they are in nature. And what I'll do today is try and give you a little bit of a glimpse in as to how these butterflies of the soul, so to speak, might be involved in one of the key processes of mental life, that is memory. So what is memory? So here is a hard disk, as you probably all recognize, and this is one form of memory. But it's a very particular kind of memory, and let me just explain what I mean. The reason why hard disks are good for storing information is because when you make physical changes, in this case, physical changes in magnetization, that change persists. So it is a memory because when you write something to hard disk, little bits literally of magnetization are set, and those will stay for a very, very long time after you have set it. And this is one form that people have considered for memory in humans and in other animals, and that is the persistence of structures such as the synapse in the brain, which can last for a long time, but as has now become clear, is perhaps not the best answer, because they are, sorry, let me go back. because synapses tend to be actually quite fragile and they turn over over a relatively short time. Some will last for a long time, but not all. So this is not the answer, the only answer for memory. Another form of memory, which you probably are aware of, is RAM, DRAM. And this is what it looks like in a computer. And the way this works is that it works through refreshing. That is, you store some information on it. After some while, the information literally discharges, stored in the equivalent of little capacitors. And so you get this discharging curve. And then it is refreshed. Excuse me. Sorry about that. So then it gets discharged, and then it can be refreshed, and therefore you can store the memory indefinitely. And something quite similar happens with neurons, or is believed to happen with little circuits of neurons, where the activity of the neurons goes around in a circuit such that it's not one neuron that's storing the information, but the fact that the memory is continually refreshed as it cycles through the circuit. So this is called reverberation memory. Okay, here's another kind of memory, and those of you who are doing computer logic will recognize this as a couple of NAND gates wired together in a bistable circuit. And what does that mean? That means that you can store, and I won't go into the logic of it, but you can store information in these little circuits such that the information will be retained. In other words, the state of the circuit will be retained as long as power is delivered. And this is through a phenomenon called bistability, which I will tell you about somewhat more in a little while. And this is the neuronal equivalent of it. Circuits, but not implemented through physical logic gates, but implemented through chemical logic gates. And we will look at that also in some detail. So let's now come to one of the crucial things which I want to get across, which is that computation in the brain occurs across scales and across levels. So and it's a huge mistake to focus all your attention at just one level, though, you know, when one gets specialized in research, one tends to focus a bit. But the functioning of the brain is so interlevel, interrelated, that focusing on one level, you're almost certain to make mistakes. So as we shall see, information is stored, memories are formed, all the way from molecules to the level of behavior and everything in between. And it is the flow of information between these levels that is crucial for what we think of as memory. So let's just spend a couple of moments going through the scales of information in the brain. So here's a brain, and this is about 15 centimeters long. It's 1.5 liters roughly. It's, you know, it's what's in your head. And compared to this, let's now zoom in a little bit and look at roughly the size of a neuron, which is this little spot over here in the corner. That's roughly the size of a neuron. And we're going to look at a neuron over here. And this is a computer model of a Purkinje neuron. And Shyam will be amused to know that I stole this model from Eric Tushuta, with whom he worked, this Purkinje cell model. But this is a very faithful geometrical reconstruction. So this was the 0.5 millimeter that you saw against the brain there. And now this shows 0.5 millimeter in size. And now we'll zoom into a small patch of the dendrites over here, and about 5 microns in size. And you can sort of see the outlines of a cube. And the structure in yellow is the dendrite. On the dendrite, you see many little mushroom-shaped things poking out. These are called dendritic spines. They're very, very important for memory. On these dendritic spines, there are these red dots, which are the synapses. And the synapses are formed between the dendrite, or to be more accurate, the dendritic spines, and axons that happen to make their way through the volume and touch it. And you should be very conscious of how utterly densely packed is the brain. So in this small volume, I've forgotten the exact number, but there's hundreds and hundreds of these axons. There's hundreds of dendrites. There's thousands and thousands of dendritic spines and synapses. It's all just jam-packed, and all this is just 5 microns. But let's go in even further. Let's go down to the size of one of these synapses to see what's happening there. So here is a reconstruction. There's not a picture. We don't yet have microscopes that can do this. This is a reconstruction of one synapse, one synaptic bouton to be precise, the presynaptic side. This is where the synaptic release occurs down here. And now you can start to see the individual protein molecules, various fibrils. You can see some microtubules. You can see a whole bunch of vesicles floating around. You can see some actin filaments. You can see some membrane proteins. So now we're talking about the computational elements in the brain right down to the level of molecules. And as I said before, and I reiterate, it is the crosstalk between all of these levels that is crucial for functions such as memory. So how do we form memories from all of this? So what I'm going to do is I'm going to work through an example based on something you're probably all very, very familiar with, which is Pavlovian condition. And here's a picture of a great man, Ivan Pavlov. And he was actually interested in digestion, or in the enzymatic properties of salivary enzymes. And but he was a good scientist, so he noticed something very interesting. So he had a bunch of dogs from whom he collected saliva. And since his was a well-organized lab, every day at a certain time, a bell would ring, and that would be a signal for the lab staff to go around giving the dogs food. That's the unconditioned stimulus. And when a dog sees food, what does it do? It salivates. Very, very straightforward. So he was recording all of this, but at the same time, remember, a bell would ring. And initially, the bell is pretty meaningless, right? The dog hears a bell, that has no particular interest to it. But after a number of associations, and association is the key word here, after a number of associations of the bell with the food, the dog started to make some kind of connection between them, to the point where it formed the conditioned response, and eventually it would salivate in response to the bell alone. And this is what we call classical conditioning, one of the earliest, very rigorously defined forms of memory. So now there's a memory formed between the occurrence of the bell and the presentation of the food, the conditioned stimulus and the unconditioned stimulus, and then there's the response. So this is a standard form of memory, and let's just do a little thought experiment. Yeah, here I'm going to make a cartoon dog, and this cartoon dog has just 3 neurons, which is of course grave injustice. Dogs have a lot more neurons than 3. But this cartoon dog has a neuron, and one of the neurons, this one in green, is the food neuron. It responds to the food, and what does it do? It causes saliva to be secreted once food is seen. Fine. So this is the unconditioned stimulus pathway, right? The unconditioned stimulus and the unconditioned response. Now, let's suppose that we have somewhere in this dog, we have another neuron that responds to the bell. And just as a control, as a good scientist, one should always have a control. Let's have another neuron which responds to light. And both the bell neuron and the light neuron are connected to the food neuron, but very weakly. Yeah? Now, what happens is during the training, the bell neuron is always associated with the food neuron, right? The bell and the food are always associated. And so this connection, this synapse between the bell neuron and the food neuron is repeatedly active, both on the presynaptic side and the postsynaptic side. In other words, there's an association between these. And we'll see in a little while how repeated association can cause changes in this synapse, in this connection. And these changes are such that now the effect of the bell neuron becomes strong, in fact, so strong that the bell neuron on its own can trigger the saliva. So the bell neuron goes off, the synapse is strong, it causes a large effect in the food neuron, and now the food neuron is active, and that causes saliva to be secreted. In contrast, the light neuron, which is not associated in time, does not get strengthened. The synapse remains weak, and so you do not learn to associate light. So this is a complete cartoon, right? Real brains are far more complicated. And yet, here we have effects happening, and I stress, effects happening all the way from the molecules, which are right down there in the synapse, through to the subcellular regions, the branches, the cells where this is connected to the whole cell, to the entire behavior, the circuitry and the behavior of the animal as a whole. So we've gone from molecules to behavior in this very cartoon schematized way. And so this is, just keep this in mind as a kind of illustration of of how one makes this bridge between molecules all the way to behavior and all the levels in between. Okay, with that, let's spend a little time looking around for memory, looking at different aspects of it. Here's a picture of what he was then, a young man by the name of Henry Molaison, who had the misfortune of having intractable epilepsy. And to address this, Scoville and Milner, Scoville was the surgeon, he made a large lesion. He removed, surgically removed a large part of what we now are, well, it was known then as the hippocampus, but what we now know is crucial for memory. So he removed the hippocampus from the brain of Henry Molaison, and this cured the epilepsy. It did that. But it did something else, which was quite tragic, because it removed his ability to form new memories, and not just any old memories, but new memories of what are called the declarative kind. So let me give you an example. Oops. So supposing supposing you're the doctor, and this actually happened. Yeah, the doctor comes in and introduces himself to the patient, and he has to do this every single day because the patient has no new recollection of this doctor. But if the doctor asks him, okay, who is the president, or who won this baseball game? He was American. Who won this baseball game before you had your surgery? Henry Molaison would remember that. So he could not form new memories, but he could recall old memories. And there were even weirder things, you know, for example. So there were other weird things that happened. For example, Henry Molaison was unable to recollect the name of the doctor, but another colleague came by, and when he was shaking hands, he stuck a pin in the hand of Henry Molaison. And of course, Henry did not remember this. But when the fellow came in again, a few minutes later, after he'd completely forgotten about him, he would refuse to shake his hand. So, and other things, he could learn, for example, he could learn to draw with his left hand, even though he had no recollection of ever having done the learning. So these are the kinds of interesting things about where different kinds of memory are stored. And so the declarative kinds of memory are stored in the hippocampus. Here's another example. This is a very unusual patient of a great Russian neurologist, Alexander Luria. He's written a little book about him, Mind of a Mnemonist. And so this gentleman had a perfect memory, and you might think this is wonderful. But actually, the poor fellow, this patient, had a really terrible time of it. And this is just one example of the deficits which the patient had. So here's a series of numbers. Now, what Alexander Luria used to do was he would give the fellow, the patient, a series of a table of numbers. And then a year later, 5 years later, 10 years later, even 15 years later, he would ask him to recall that table. And the gentleman always could. He simply remembered everything, even things that had happened all those years ago. But the way he remembered was very interesting. So now look at this series of numbers. Yeah, you and I look at this and you say 1, 2, 3, 4, 2, 3, 4, 5. And so on, you immediately see that this is not just any old table of numbers. This is an organized set of numbers which go in a certain sequence. The patient didn't know this. He didn't recognize it. He simply memorized everything. So when we form memories, we are at the same time doing an equally important, maybe an even more important thing, which is we are interpreting, we're synthesizing, we're understanding what those memories mean. And if we don't do that, if we just store information, then actually we are very, very severely impaired. We're very, very limited in what we can do. And it's the ability to interpret at the same time as we are forming memories is actually a crucial part of how humans think, I would argue. I won't go into the other interesting things about this patient. He had very, very strong synesthesia. That is, he could mix up different kinds of sensory modalities. Different numbers, for example, had different colors and other attributes associated with them. But I will move on. So, so our friend Henry Molaison, he had, as I said, he had the opposite kind of problem, very bad memory. But as we discussed, he lost his short-term memory, but could recall old things. And he could still learn to form new procedural memories, such as learning to draw. Now, let's spend a little bit more time on the hippocampus, which was affected in Henry Molaison's case. So in humans, the hippocampus is this little red structure. I hope you can see the rotating skull over here. So it's bilaterally symmetric. And it's not such a large structure. It has been studied a huge amount, and it has a very nice property, especially in rodents, but there's something similar, but of course more complicated in humans, which is that it forms what are called place cells. And to put it very simply, if you record from a cell in the brain of in the hippocampus of a mouse or a rat, this cell will be active at a certain region in place. So, for example, if you put the animal in an arena, in a large, let's say 3 meter by 3 meter square to run around in, the neuron will be active in a certain subset of that. Or if you put the animal on a linear track, the neuron indicated in these different colors, different neurons will be active in different parts of space. And the interesting thing is the animal can be taken away and come back the next day. And again, the same neurons will be active in the same places. So and there's a lot of other, a huge amount of work has gone on this. Nobel Prize for biology and medicine was given a few years ago to the Mosers, among others, for studying these properties of hippocampal cells, the formation of memory of place. So this is where one spends a lot of time, and the field as a whole has spent a lot of time looking for memory in the hippocampus, but other regions also, like the amygdala, which is responsible for emotional memory. That's at the systems level. If you want to go down a little bit, circuit level, one looks at recurrent and associative networks and kinds of reinforcement. There is a whole lot of stuff that happens at the cell and circuit level, connectivity changes, excitability changes, how the genes are altered. And then at the synapse level, this is if you remember the discussion on the Pavlovian conditioning, how the conductance changes of the receptors there, how much neurotransmitter is released. And you can go right down to molecular level. And so what I'm going to do today is I'm going to spend a little time discussing a couple of levels, the area level, and I'll talk a little bit of the molecular level. So let me just go straight there. So area level. So what we're going to do is I'm going to just give you a very, very brief glimpse of our work, where we look at the activity of the brain, in this case of the hippocampus. So this is the mouse hippocampus, which we image using little windows that we place in the skull over the brain. And these little windows are, through these little windows, we shine light using a technique called 2-photon imaging. I won't go into the details. It allows one to look relatively deep into the brain tissue, because brain tissue is not normally transparent, but using this technique, you can see deep. And optically, one can monitor brain activity through special dyes, through special even genetic constructs, which will be brighter when the cell is active. And I won't go into details. Suffice it to say, this is an amazingly powerful technique. And although the images I'll show you have only looked at a few, 50 to 100 cells, nowadays one can look at 10,000 or more cells at the same time using such techniques. So we've built 2-photon microscopes to do this. In fact, the first generation microscope was built by students on their own design. And of course, if you're going to study learning, you have to train the animals. So there'd be something called trace conditioning, which is actually very, very similar to Pavlovian conditioning. There's a small gap. So there's a CS, a sound, just like the bell of the dog. Then there's a gap. And there's something called the US, the unconditioned stimulus. In the case of the dog, it was the food. In this case, it is a little puff of air that goes towards the eye of the animal, so the animal blinks. And just like in the case of the dog, the animal learns to anticipate the second stimulus, and it blinks even before the air puff comes. So let me show you. So here is one of the earliest videos we took of brain activity while the animal was learning. The little bright spots are neurons. And if you look closely, you can see them kind of flickering on and off. And this flickering on and off is brain activity. It is the computations that the brain is doing while the animal is experiencing sensations and learning things. Okay, so we study many things. We study formational memories. We study how they start representing information. We ask what happens if the animal has to remember more than one thing. How can memories coexist? We ask how memories can be forgotten. All of these things are part of trying to understand memory. But I'll just look at one of these things, if my slides will do me the favor going forward. So the formation of memory is something that was studied by the students who built the microscope. This is Mehrab Modi and Ashish Dhawale. And so Ashish is now a faculty member here at ISC. And Mehrab will hopefully soon also become a faculty member somewhere. And what they found was that before training, the activity of cells, which is indicated over here. So here they have 6 cells. Each cell they recorded multiple trials. They've just shown 3 trials over here. And before learning, the activity was all over the place. But after learning, as you can see, the activity is very strong and it is almost perfectly lined up for each of the cells. And the interesting thing, the really interesting thing is that if you now compare this with the timing of the stimuli, the activity forms kind of a sequence that goes from the start, from the sound to the end, which is the air puff stimulus. So a structure of activity is forming as the animal learns something. And I think this is really very fascinating. Okay, so that was the systems level. Now I'm going to switch to molecular level. The huge mess of material that is present in every single one of your synapses. Just to put it in context, you have roughly 100 billion neurons in your brain. And each of those neurons has anywhere from 1,000 to 100,000 synapses. So there's an awful lot of these synapses. Yeah? And each one of them is jam-packed with, as you can see, all this potential computational machinery. So let's look at how it works. So this is a very, very old model I made many years ago as a postdoc. And it's still, as you can see, even back then it was pretty complicated. Now our models are even more complicated. And I won't go into the fun bit about how one builds the models, gets data. That's something we can discuss later. But this is what the kind of model that we can make more recently, where each one of these little dots, each one of these little stubs is a synapse, is a dendritic spine. Each one of those has all of the signaling, all of that, not necessarily that complicated, but a huge amount of complicated chemistry in every single one of these. The dendrites, all along them, have all this chemistry happening. There's all the electrical activity. And this is because this is what real cells do. Yeah. So nowadays we can make computer models that actually have all of these details happening at the same time. And in this particular case, we gave a sequence of input. And the outcome of it was that when the input was in a sequence, you got a whole section of dendrite lighting up. But if it was not in sequence, it did not light up, which is you see on the right side. So here the input is not in sequence, it doesn't light up. So these are just some of the computations you can do through the signaling, through the chemistry. But let's go to this key question of bistability. So bistability is actually a very simple concept. Yeah, if you think of some kind of a surface in which, and you have a marble there, and the surface is shaped like this, the marble will be either in one energy minimum or well, either that one or that one. And there'll be some kind of an unstable position between the two wells, where if you push it hard enough, it'll go up and then it'll go down to the other one, right? So this is called bistable because while the marble is in one state, you can move it a little bit, but it'll come back to the lower state. If it's in this state, you can move it a little bit, but it'll come back. And it takes a really strong push to get it to the other side. So this is mathematically identical to something that can be done through the chemical signals. And let me tell you a little bit about that. So supposing we have, as our chemical circuit, a very simple chemical circuit, we have a system which has a molecule A, which activates B, and a molecule B, which activates A. It's a little feedback circuit. So now you can just imagine that in the resting state, A is off, B is off, but now you give it a very strong stimulus. So A becomes on. Because A is on, it turns B on, B becomes on. And now because B is on, A becomes on as well. And so this circuit stays in the active state. Now let's separate it out and think in terms of our synapse, where there's hundreds, thousands of these little molecules floating around. So that's what I tried to represent over here. So here you have a bunch of A's, which are the triangles, and a bunch of B's, which are the little octagonal shapes. And when they become stimulated, they all become active. So now let's put these into these kind of mushroom-shaped things are meant to be these dendritic spines. Let's put them into the dendritic spines. And now let's factor in a really crucial part of how the brain works. and the body works, which is that proteins have a short lifetime. All these molecules will only last for, say, a few minutes to a few days. So what happens then is, you would think would be disastrous, right? You have your memory formed, but now some of the molecules get degraded. And so they're degraded, so there's like a hole in this, there's gaps in this set of molecules. And if this went on, of course, you wouldn't have any memory and you wouldn't have any spine either. You wouldn't have any cells, any molecules left. But of course, the cell is alive, therefore new molecules are synthesized, and that's indicated in these lighter colors. Now, the crucial thing is that these new molecules don't, quote unquote, know. They're not part of this memory, right? They're fresh molecules, they're naive. But because there's overwhelming amount of activity over here, just like a small perturbation on that energy well diagram, this formation of new molecules is like a small shift of the marble, and then it comes back to its steady state. And its steady state in this case is one of activity, because most of the molecules are active, and therefore they turn these molecules on. And then they become part of the memory, and you can keep losing molecules and keep getting fresh ones, and yet you will not lose the memory. And in some ways, this is a lot like the oral traditions, right? The oral traditions of teaching, where the guru would teach the disciples something. Of course, there was writing, but a lot of teaching was done verbally. And through memorization, it didn't matter then, in due course the guru passed on, but then the younger disciples themselves became teachers, and they passed on the story. So this is how oral traditions are passed on. And it'd be kind of interesting to think that something similar is happening with the molecules in your brain, passing on the tradition, so to speak, that make up your memories. Okay, so now let me just close this loop, right? We looked at memory at the level of the entire animal and the circuit when we were talking about Pavlovian conditioning. We looked at memory at the level of many cells performing some activity in a very particular way. That is a somewhat more complicated version of the bell and food connected activity that we have thought of in this example of Pavlovian conditioning. We skipped over a lot of interesting things that are happening at the cellular level, and we sort of focused at the molecules here at the synapse. And remember, we can now make the synapses, we've now seen through the bistability, how we can make synapses be either weak or strong, simply based on the level of activity that those synapses, that the molecules in those synapses express. So in the weak synapse, it might be similar to a case where all the molecules are in this state. So they have very little synaptic transmission. And in a strong synapse, all the molecules might be in this state, so they have strong transmission. And so here we have a bridge, all the way from molecular stuff happening here, to circuit level stuff, to network level stuff, and eventually being to behavior. So I hope I've given you a perspective of how all of these levels may come together. So now what I'm going to do is I'm going to get a little bit futuristic. Yeah, it's a topic that I'm sure you're all very engaged with, thinking about artificial neural networks, AIs, as they're now popularly known. And I'll just give you a couple of examples of what one can do, and I'm going to try and link this to the concept of memory. So now let me at the outset state some of the limitations from biologists' point of view for neural networks, artificial neural networks. First of all, they're unrealistic. Yeah, we call them neural networks because once upon a time, some very brilliant mathematicians and computational neuroscientists said, how can we simplify, really simplify and abstract out the properties of a neuron? And they came up with what we'll see in a moment as an artificial neural network unit. So they're unrealistic. They throw away a lot of this detail. For example, all of this lovely molecular computation is absolutely nowhere to be seen in an artificial neural network. They're unrealistic, but they're very instructive because when you abstract things out, you can start to make theories, to make mathematical and computational models of this. And this can be very interesting for thinking about what the real brain is doing. Not only are they instructive, they're useful. As you know, nowadays more and more things are turning out to be of value from artificial intelligence neural networks of those kinds. And one of the fundamental operations that these things can do is classification. And one of the fundamental commonalities between our artificial neural networks and the ones that we were just talking about is that information is stored in the synapses. The connections between cells is the site of storage of information. Now, in the brain, as in biology, that's not the only place where information is stored, but in neural networks, that is pretty much where all of the information is stored. And another very interesting commonality, which is at the other level, at sort of the behavioral level, if you like, is that you and I, we learn by example. Yeah, we see things, we see a few illustrations of something, and then we know we understand the problem, we know how to do it. Now, in a program, if you write a computer program, you can't do that. You have to be very explicit and tell the computer, this is the variable, these are the steps that you have to follow to do it. Yeah. The computer does not learn by example. But one of the nice things about neural networks is that they do learn by example. You can train them by giving them a lot of cases and say, yes, this was the correct output. No, this was not the correct output. And you do this enough times with enough inputs, they quote unquote learn. All their synapses through various learning rules get altered to allow the network to behave in a manner which looks like it has learned something. So now let's have a little time thinking about this. Yeah. So here's an abstract neuronal model. And all we have here are, we have a bunch of inputs. Each input converges to the neuron, which is just a summation device, with a certain weight. And so you can sort of think of these weights and these as forming like synapses, these inputs and weights. So the input is multiplied by a weight, and then that is added up. Input 2 multiplied by a weight, added up, and so on. So that's the mathematical form of this. And then all of these sums are sort of put together through some kind of nonlinearity to give you the output activity of the cell, of the unit. It shouldn't be called a cell, it's way too simple, right? So this is so simple, and yet it works so well in all our artificial neural networks. So you can put them together in many layers. You have your input layer, you have a hidden layer, typically you can have many hidden layers. In fact, one of the big things in what are called deep networks is they're deep in terms of how many hidden layers they have. And for decades, compute power and the limitations on training sets meant that you could only have 1 or 2 hidden layers. It takes a lot of training to do it. But then along came the monsters, Google and Facebook and all of these terrifying super companies. And they had the computing power and they had the data sets. So they could do deep training, which has many, many layers. We won't go there we won't go there. Yeah, there's a whole history behind that. But let me show you something that can be done with even this very simple network. This was something which was done by Terry Sejnowski many years ago. So let's take a simple neural network with all these inputs, all of these arrows, each one of which represents a weight. And we're going to train it to do a very difficult thing, which is to pronounce English correctly. And English, as you all know, is really bizarre in how it comes up with spellings, right? Unlike most of the Indian languages where the alphabet is phonetic, you look at it, you know how to pronounce it. That's not the case in English. So let's take a word like neuroscience, which has got all these interesting letters, some very confusing. And what we want to do is we wanted to train it to pronounce it like neuroscience, neuroscience. Yeah. How do you get the neural network to learn this? Well, you simply give it a lot of examples. You give it an example of neuroscience, each letter being connected to one of these inputs. You train it. By training, I mean you see what sounds it generates, and you say, no, that was correct. No, that was good. That was bad. That was medium. And then through a technique called back propagation, the information goes back. All these little synaptic weights are changed. And this is how the knowledge, so to speak, the training happens through many, many examples of words, their spelling, and their actual pronunciation. And in the end, the neural network could learn to pronounce reasonably well. I mean, not as well as modern speech synthesizers, but pretty well. So now fast forward 40, 50 years, and here's I'll just give you a couple of brief glimpses about what you can do with artificial intelligence. Here's an example of a diagnosis thing. So many neural networks have been trained to look for diabetic retinopathy, which is a condition where your vision gets really messed up because of diabetes. So these guys, they trained the data on a fair number of patients, and they had 2 validation data sets, the test data sets. And what they did was they also had information, general health information about these patients. So they had retina scans, which is what you're seeing over here. And they had general health information. And so after this, something remarkable happened. The neural network could look at one of these retina scans and predict the following. It could predict the age within 3 years. It could predict the gender to within 97% accuracy. It could predict whether the person smoked with 70% accuracy. It could predict their hemoglobin type. It could predict their blood pressure, and could predict whether or not they'd had a heart attack. All by looking at the blood vessels in the retina. Okay, so this is an example of how you train it on lots and lots of things, and it sees subtle patterns in this that can now be used for diagnosis. Here's another example that many of you may have seen. This is something called a generative neural network. So what you have is you have a few source faces, just ordinary pictures, and you train up the network to generate faces. And I won't go into how it does it. It's a whole field in itself. And then you take sort of sample style faces, and you ask it generate new faces. So the only real faces in here are these guys on top, yeah, and a few sample faces here to the left, and it generates everybody else. All of the faces down here are synthetically generated, and they all look totally convincing. And to me, the really mind-boggling thing is, you know, look, here's a girl. There's no mustaches anywhere to be seen, and yet here you have two guys with perfectly decent-looking mustaches, yeah, and they look reasonable. Here you have an adult. There's one kid sample, and you get a completely different looking kid. Yeah? So this is all what you can do with neural networks. Now, why did I spend all this time talking about neural networks? Yeah. Okay, one more thing. This process of training is now big, heavy computing, right? This is just one of the tensor processing units that Google has. This thing occupies a giant room. I don't know how many megawatts it consumes. And you have to remember that this is just doing one tiny fraction of what you do in your brain, which fits in there and takes about as much power as a small light bulb. Okay, so memory and AI. So now, the reason why I bring this up is that AI is, one way of thinking of it is a very, very inefficient way of using huge amounts of memory, huge amounts of training, and it can just begin to solve some real world problems. And we do this far more efficiently. But there's a rider to this. So far, we do this far more efficiently. So what's happening, as I'm sure you all know, that in very specific domains, very specific problems, AI can be trained to do better than humans. And I don't know about you, but I lived through this revolution, and it is scary as hell. Pardon my French. But yeah, so for general things, we're still on top. But for many, many specific problems, AI is really, really catching up. Anyway, you are going to have a very interesting world, all of you young people. Okay, and the key thing is it is using memory. It is using synapses. That's the same kind of thing that we've been discussing that the human brain has so much of. Okay, so let me wrap up. So where do you look for memory? And just to recap, you have to look for memory at all these scales of function, from the whole brain to the cells to the subcellular regions to the molecules. You form memories at all of these levels. We've seen how you can use cellular and synaptic things and molecular events to form behavioral changes in this cartoon of a dog, of a Pavlovian condition. Yeah. And all of this is happening through the functioning, the connections, the synaptic events happening in these beautiful, beautiful neurons, the butterflies of the soul. So with that, I will say thank you to all of you. I'll say thank you to the many people in my lab over the years, the many people who funded me, and thank you for listening.
Great, Dr. Bhalla, thanks so much for the wonderful talk. If you have any questions, you can ask those questions to Dr. Bhalla directly. I'll start with mine. So Dr. Bhalla, you spoke about the model, right? The electrical model, the compartmental model. Would you mind talking more about that? I mean, which neuron is it?
Oh, the video I showed?
The one with a lot of dendritic spines on it.
Oh, okay, that is a hippocampal neuron. So this is a standard hippocampal pyramidal neuron. The model has only about 5,000 synapses, although it looks like a lot of them. I didn't have the compute capacity to do even more. So that's about half the number of cells of synapses it should really have. It has, I think, about 1,000 morphologically defined compartments. And every single one of those has lots of chemistry going on in it.
All right, yeah, thank you. So one more question before I give chance for others. So you spoke about neurons and their important role in memory, right? So one of the most underappreciated structures, or one of the most underappreciated components in neuroscience are astrocytes and glial cells, right? So do you think they're also...
Sorry, what? The underappreciated structures are what?
Astrocytes and glial cells.
Oh,
right?
yeah, absolutely. Yeah.
So do you think they're also involved in memory? Would you mind,
of course they are, absolutely. Of course, yes. I mean, you're sort of going into more depth than I did, which is a good thing. So let me put it this way. Every synapse, somewhere I will have a schematic of a synapse. Let's see, where do I have a schematic of a synapse? Okay, here we have a schematic of a synapse, right? I've drawn it as what's called a bipartite thing. It's got a presynaptic side and a postsynaptic side. Actually, in your brain, every synapse is a tripartite synapse. It's got the presynaptic cell, presynaptic neuron, it's got the postsynaptic neuron, and it's got a wrapper, so to speak, of a glial cell. Yeah, and the traditional view has been that these glia are just acting like kind of housekeeping. They're making sure that the cells have enough fuel, that they're all the waste products are gotten rid of, that kind of thing. But no, they are actually very, very closely involved in the signaling pathways. They can themselves transmit slow calcium events, slow calcium waves that go long distances. They're very important in managing the metabolism, which is crucial for keeping this going. So yeah, they do an awful lot of stuff. I mean, I can go into more depth, but I think that one should just appreciate that I've talked about neurons and synapses, but there's another major player in that.
All right, thank you, Dr. Bhalla. Shivani?
Hi, am I audible?
Hi, yes. Shivani, yes, we can hear you.
Thank you. Thanks so much for that talk, Dr. Bhalla. I have a question. I hope it's not too irrelevant. I was very intrigued by your explanation of how proteins need to be built constantly to maintain memory. And I'm thinking from an evolutionary perspective, the ultimate causes for needing to, for doing that are understandable. But as we imagine, maybe not so right away, but the reliance on memory by human populations because of technology is going down. Do you think that is likely to affect, from an evolutionary point, how such proteins may continue to maybe be the energetic cost and how that may affect evolution of memory retention?
Yeah, I mean, the technological age is just an eye blink in evolutionary time. I mean, if it goes, things are changing so fast now that we are still working with the brains that evolved 100,000 years ago, and there's not going to be very much evolution in the very short time as all this change happens. I mean, looking forward is a very dangerous exercise. Some thinkers, Kurzweil and others, have speculated there's going to be a technological singularity where basically machines get faster and faster, and then advances infinitely fast, I mean, almost like a black hole of information. I don't think so, but surely the profile of evolutionary pressures on our brains is going to change dramatically over the coming years. I mean, I suspect that more than the reliance on external memory sources, we're going to have to deal with things like implants of various kinds, which will interface with the brain. We're already talking about brain-machine interfaces, brain-mind interfaces, right? Brain-machine interfaces. So I think that is where, in long terms, such kinds of things would happen. But we're squarely in the realm of science fiction. I mean, all of you are living in a science fiction world now.
Thank you.
Shreya, you had a question?
Yeah, so I had a few, but one of them was kind of related to Professor Shivani's question, where like the fact that the digital age is kind of reducing people's memory, so to speak. But I was wondering if that is like, it doesn't have something to do with memory formation as much as it is retention. And my question was based on repression of memory. So like it's sort of correlated with that.
Mm-hmm. Yeah.
So
So,
it's just basically, how would you explain repression with your like models?
Well, no, you've actually stated it very well. Repression is a form of memory. That is, it's not simply forgetting. It is actively working through circuits, through patterns of activity to prevent certain behavioral outcomes, even though the memory is there somewhere, right? So it's got learning to put a lid on something. Now, I really want to stress that, we have this impression of the digital age weakening our memory. I don't think that there's any really strong evidence for that yet. There's effects that seem to be happening in younger generations of attention span, which is a natural outcome of being able to monitor so many things at the same time. That's a psychological thing, though. That's not the same thing as a change in the fundamental substrates of memory. And I think that if you go ahead, and since I haven't read anything to the contrary, if you go ahead and do tests on simple memory capacity, the thing of learning a 5-digit number and another 5-digit number and seeing how far back you can remember them, I don't think that has changed. And I don't see why that would change. If anything, the sheer amount of information which young people are exposed to, they're doing an excellent job of filtering important stuff, salient stuff, and focusing on relevant stuff, by whichever criteria they choose to do so. But it's precisely the thing that failed. And if you remember that Russian patient who had a perfect memory, he was not able to do this filtering. And I think it's important that that people are responding to a barrage of information by being very good at filtering. So I think that we're still pretty good.
Satvika?
Yes, thank you. I really enjoyed the talk and it was very, very insightful. I just wanted to ask about when traumatic experiences have been experienced by an individual, and oftentimes they entirely forget that the traumatic experience has happened, and there's that kind of repression of the memory. What exactly is the process behind that, and how exactly does that work?
Well, I can't tell you exactly. I mean, this is still a research topic. I mean, the broad outlines are very well understood, which is that certain parts of the brain, in this case, the amygdala, is particularly responsive to emotional stimuli. And for example, a post-traumatic stress disorder as a negative emotional stimulus clearly has a major representation in the amygdala. But also positive emotions also get strongly represented in the amygdala. So there's some level of specialization about which brain regions recall things. And these are not a problem. It only becomes a problem when the memory becomes overwhelming, when it generalizes too much. So you're in a context which is actually non-threatening. And despite it being non-threatening, you are triggered to recall something traumatic. And this is a well understood, reasonably well understood phenomenon of overgeneralization. And so then one, as you have said, one comes to terms with what was the original trigger. One tries to, there are various kinds of therapies that look at how one can get people to recall the trigger and slowly recognize to sort of reduce the level of generalization. And it can be very difficult because the brain is designed to be very efficient at catching dangerous cues. I mean, it's a survival thing. And so, it's unfortunate that such events, in fact, utilize very, very powerful, efficient mechanisms in the brain.
Thank you so much. Thank you.
Dr. Bhalla, you have time for one more question. There's one student interested in asking a question.
Sure.
Katan, please.
Yes, firstly, thank you for a wonderful talk. My question was that usually we hear that a person's memory retention kind of reduces as they age. Mm-hmm. So could it be like down to the molecular level of proteins, and because you said that proteins and the constant requirement is needed for memory retention, then could it be down to that molecular level of why as a person? But at the same time, there's also neuroplasticity, so it's like kind of contradicting.
No, but you sort of stated something which I think is a key message for the talk, which is that all these events, memory is not something that you can just focus on one level. You have to look at all of these levels. So it's very clear that there's huge neuronal loss, especially in certain kinds of dementia. What's really amazing is how much functionality we retain despite a huge amount of neuronal loss. But at some point, it goes beyond the point where the circuit can make up for it. And at the same time as there's neuronal loss happening, there's certainly changes in the metabolism, the efficiency of forming new synapses. All of these go downhill with age. So it's a multi-level process. And yeah, I mean, there's a huge amount of very important research going on to try to understand which are the sort of the weak links in the chain. What are the metabolic, the molecular changes that lead to the neuronal loss in the first place? What can we do to perhaps, quote unquote, rejuvenate the neurons, maybe give them more flexible plastic synapses? So all of these changes happen across these levels.
Right. Thank you so much.
I don't think we have any further questions. Thank you, Dr. Bhalla, for the wonderful talk. Thanks everyone for joining. If I can, Shreya, yes, sorry.
Yeah, sorry, I just had another, this is a discussion question, I guess. But in your slide, when you mentioned biologists' criticisms to neural networks, one of them was the fact that they have to learn, they have to be taught everything. They get information through learning, but aren't humans also like that? So, or is there something that we don't learn and we know before?
No. So I maybe wasn't so clear. It's not that, no, it's actually similarity. We in neural networks learn by example. The difference is how many examples do we need? Yeah, so humans will learn from a few examples. Yeah, but it would take you a million lifetimes to try all the examples that Google tries for its big AIs. GPT-3 has, I don't know how many billion parameters, is it billion trillion parameters? It's got some completely insane numbers of values that it stores. And the training sets that are so huge, the power costs alone for doing the training is the order of tens of millions of dollars. Just the energy costs for training this thing. Yeah, we don't have time for that. We have to survive. We have to, we have to propagate the species. So brains are very, very, very efficient at learning with very few training sets, very few examples.
Right, that makes sense. Thank you.
All right, thank you, Dr. Bhalla, for the wonderful talk, and thanks everyone for joining.
Thank you all. It's been a great pleasure, and thank you for the very interesting questions.